Good day, class. I want to welcome you once again to term 3. I'm quite sure for grade 12, you have got a story to tell. Life science is here at Metric Hub. I want to really assure you that this is an educational gateway towards your distinctions in life sciences. If you know anything that you can ever do, please follow Metric Hub and get yourself tuned and connected each and every time. I'm here for you. Here, this is the best place to be. Otherwise, you believe that Metric Hub, that is where you find all your resources packed. Genetic guru. Don't worry, we are here for you. We can simplify, explain, calculate, and all questions that you think of in life sciences. We can really dissect and present them clearly and well done for you. Let's connect right now. Share to friends, share to everybody, and we can develop a good community of young intellects without wasting any time. This is 2025 Free State paper, which I want to believe that is going to give us a good reference of revision towards term 3 end of term exams. Also note that Metric Hub has got lessons from grade 8 to 12. Share to your brothers, share to your sisters, and everybody else out there who is stranded. We are here to actually give the best rescue. We have got instructions which I'm quite emphasizing for grade 12 with due respect. We no longer have time to make errors. Please make sure, read instructions and apply them accordingly before doing any mistakes. Uh, Question number 1, we just wanna go straight to the paper and rush as much as possible, of course, with comprehensive explanations for all possible answers and situations that we are going to do today. And then the first question reads, which of the following characteristics applies to bipedal organisms? I'm sure this is evolution, and for your own information, we have got exclusive videos that are there for you on Metric Hub channel and also other question papers that you can refer. Bipedal, we are saying walking on two legs. That is, what is the meaning of bipedal walking on two legs? On two legs is what you mean by bipedal. So which of the following characteristics applies to bipedal organisms that walk on two legs? What is it that is the best, which is actually a characteristic of them? More backward position of the foramen magnum. The foramen magnum, if you can check, we are saying the opening at the back of the skull, which is actually connecting to the spinal cord. It is not correct, short and wide pelvis. If you can check organisms or individuals, or we talk about uh, evolution and science, it is known that bipedal organisms have got a short, broad, wide pelvis, which is actually designed to sustain the upper body weight. So the answer is B. Actually, B defines a bipedal organism. The pelvis is wider and short. We shall explain this again as we go, but that's what we need to understand. Which of the following shows the process of DNA replication in correct sequence? DNA replication, a process by which DNA makes a copy of its own or of itself, which is a prior process before cell division, that DNA must be multiplied. So the correct sequence in the process of the DNA replication, we need to understand that DNA double helix structure, it unwinds, it unzips, complementary nucleotides pair up, two DNA molecules are formed, which we can actually actually see that B DNA structure, it unwinds. We have got the double helix, the double strands, they unzip. And of course, we have complementary nucleotides pair up. The complementary pairing of bases, it occurs, and two DNA molecules are formed, which is the correct sequence in terms of DNA's replication, DNA making a copy of itself. A trait that is a range of intermediate phenotype. We said it has got a wide range, which I always want to demonstrate in a graph to say that we are looking at a trait that is having a graph that runs like in this order, in this way. We say that is called a wide range of intermediate phenotype, which we say it is starting from 0 and then it stretches in between and drops back to 0 again or to 1. What is happening is this is called a continuous variation. It's called a continuous variation, an opposite of a discontinuous. We said discontinuous variation, it has got uh, a graph that rise and fall immediately. So what happens is that we are saying it has got limited intermediate phenotypes. It is got limited, but this is called continuous. This is called continuous variation. And then, of course, we have got what is called discontinuous, which is also the opposite of it, as you can see. So it is called the correct answer is called a continuous variation. Which one of the following is the possible genotypes of the parents P1 that will result in a genotype ratio of 1:1 in the F1 generation? I'm sure if you can come here, you are meeting with the genetic guru. I want to clearly explain and show you this every time when you are going to meet them, you shall write with a smile. So what happens is that we are saying which can result the genotype, which of the possible genotypes of parents that will result in genotype ratio in F1, first filial generation, first parental generation. So we are saying if we look at this, BB and is crossed with BB, it results in a ratio of 1 only. We say that it is because this B and that B comes. I want you to just, this is revision. I want to show you all this, how it happens. We have B there and we have B there, which we say these are called the gametes that comes in here. The gametes, which of course they come together and then they give us BB. It is a 1. There is no 1:1. We take the second one. This one, it's a B and is crossed with a B, and then the gametes that comes here, we are going to have we are going to have B, and therefore we also have here there is 2B recessive, or it is homozygous B recessive, which we have got 1B there. So when the fertilization occurs, we have this one BB, and then this one it will also have BB, which we can say it is giving a ratio of 1, either if dominant B means black, it's 1:1 recessive, which is either it is white, 1:1, which we can take. And also, even this one, it will give us the same ratio as the first one. The fourth ratio is B, and is I'm sure that you must know that this will give you four possible outcomes, which we have got B, and B is crossed with B and B there. So if you can quick it out, it will give you to say we have got this one and this one. It gives us BB. We have got this one, We have got this one. It goes with this one. It gives us B, B. We take this one. It goes with that one. It will give us also B, B. We have got this one, recessive B. It goes with the recessive B, which gives us a B. So I'm saying if you look at this will give you a ratio of 3 to 1. This gives us a ratio of 3 to 1, which I will say whenever you are working within life sciences, this is going to mean if we say B is equals to black, that means to say we have got black 1, we have 2, we have 3. That is where the 3 comes from. And then this one, if it means white, therefore it would mean also to say this is what 1 white, depending on the uh, key that is there. So this is 3 to 1. This is only 1, which is the same as C again, is 1 only, because we can just have B coming as an outcome. So our answer that you should understand here is that the correct answer which comes here is that uh, we can have B, the correct answer here, which is our correct answer is this one, which is B. B is the correct answer. It gives us a ratio of 1 is to 1. I said that you can prove these questions, you can work them out. You can work them out and get them right. As you can see there, that is what is happening, or that is what you should do. So your 2 marks are gone and are correct. You understand? That is what you need to understand in terms of this. So the following statements are sources of variation. Variation, it actually causes organisms to look differently. So what we need to look at is which of the following combination is a source of phenotypical variation. We say phenotypical, the outward physical appearance of an organism, physical appearance of an organism is what we mean by phenotypical variation or phenotypical expression. So now, as you can see, we are saying the one that can affect the outward appearance of organism. I want you to understand that we can say random fertilization. Fertilization is always involved because it carries the genetic makeup. Fertilization, it has the genes of the individuals involved. It carries the genes that must be influencing the organism, which are in the nucleus of the gametes that are involved. We say that crossing over, no ways that it can never be excluded in variation, because it is the one that causes the gametes to look differently. And then random assortment of chromosomes at metaphase. I'm sure you remember that it is also a source of variation which we cannot override. Chromosomes, they actually do uh, arrange themselves at the equator during metaphase. Mutation, of course, it changes the appearance or the type of protein to be formed. So what we need to be understood is that all of them, they can actually affect or sources of genetic variation, which means A is correct. And then the diagrams below represent the pelvises and skulls of hominids. the pelvises and the skulls of hominids that we can actually need to understand the correct combination of the pelvis and the skulls, the correct combination to say they must match to say this is a bipedal, this is a quadruped, and of course, again, in terms of their hominid background. So now, what has to be understood is that if we look at this, we're saying a wide, short pelvis, it is supposed to be going together, two and we are saying B and 2 goes together. And of course, we can also understand that the quadrupedal have got they've got a very short and long pelvis. So as you look on this diagram, which one is the correct sequence of these? Skull number 3, we are saying skull number 3, it has to go with pelvis number 1. Skull number 3, this they go together. This they match, they should match. We are saying 1 and 3 must match. 1 uh 1 and 3. If we can look at this, we are saying the correct pelvis is, if you can look there. So what has to be understood is that one pelvis, we are saying skull 3, skull 2, skull 3. So pelvis number 3, it goes with skull number A, is the way I'm saying to say that in order for us to put it properly, we need to understand that these pelvises are gonna be given in a way that gonna assist us to show. Let me show it nicely here so that we can see. What is happening is that if we look at these pelvises, I'm saying skull number 3, it can go with pelvis A. This and that can go together. Pelvis A, it goes with the skull number 3. And then pelvis B, it goes with the skull number 2, which I was saying that if you look at this, it can also tell you that this one is a bipedal, which also must go together. They must match as it is like that. And then pelvis C, it goes with skull number 1. Pelvis C, it goes with the skull number 1, which we can also give to say this one, they also go together. So what is happening is the arrows that I've given identify to say skull A, it goes with pelvis number 3, which makes our answer to be what? To be C. I'm saying that is the matching that must be there to say that we see skull number 3, it goes, skull number 1, which is a quadrupedal, which is also short and long, is also going with its this and that. They go together. We say this one and this one, they go together. Same applies to say this one, it goes together with that one. That is how the matching is to take place. That is how this matching must take place, confirming our answer to be what? To be C. So that's how we can work out this. Question number next. Which of the following statements relate to Lamarck's theory of evolution? Lamarck's theory of evolution. I'm sure I remember the theory of use and disuse. Uh, theory of use and disuse, and also acquired characteristics, inheritance of acquired characteristics. That is another school of principle of Lamarck's theory, inheritance of acquired characteristics, also explaining Lamarck's theory. So now we are saying in this question number 7, which statement relates to theory of Lamarck? So acquired characteristics is inherited, which already we have said already that according to this, it's use, a theory of use and disuse, and the law of inheritance of acquired characteristic, or also use and disuse. So which is obviously making B to be correct. Theory of acquired characteristics are inherited. And then we have got refer to karyotype. We said the karyotype, it is the structure, the order, and appearance of chromosomes. It explains the karyotype. We look at the structure, we look at the order, we look at the appearance of chromosomes in an organism or in an individual species that defines the karyotype. So the karyotype represents the chromosome present in human somatic cell. One thing that we need to understand is that this question, I've also given a good video which explains the issues or questions that arises on the karyotype diagram. So let's just answer the chromosomal composition of this cell is the chromosomal composition. We are saying how many chromosomes are in this cell. It actually has, we said, one thing that we need to understand is that the gonosomes are sex chromosomes also, are called sex. They are also called sex chromosomes. And then we say the autosomes. These are any other somatic chromosomes. These are also chromosomes other than sex chromosomes. So what we are saying is that gonosomes are always known that there are two of them. So what happens is that if we look at this, we have got the chromosomal combination of this cell is, it is actually showing 45 autosomes and 2 gonosomes. Gonosomes, we have got 2 which are there, and then we have got also 45. Instead of them to be 44, we have got 3 here, which is making them to be 45. So we have got 45 autosomes. We have got 45 autosomes and 2 gonosomes, where C is correct. And then how many ova would carry the X chromosome if meiosis occurred in the above cell? How many ova will carry X chromosome if meiosis occurred in this cell? Obviously, we are saying in this cell, how many ova, it just, in this case, it will just have to say it splits, so it has got to be 1. We have got 1 in this cell because meiosis divides by 2, so we only have 1. Meiosis principles. Meiosis divides, it divides by 2, which is we have got 2 here, so we are going to divide by 2. Divided by 2 is equals to 1, which gives us the one answer that you are seeing in this case, to say 1 is what we have. So that is what happens there. And then filial, there is what is called Filial atrial fibrillation is a condition in which individuals would have an altered heart rhythm, that is the beating of the heart. So, filial atrial fibrillation is an inherited condition that follows an autosomal dominant. It follows an autosomal dominant inheritance pattern. Shaded individuals in the following pedigree have this condition. Shaded, they have got the condition. So what is given there is that by studying the pedigree diagram, it could be concluded that if we look at this, it can be concluded that if we look at this, what we can see, we are saying it is a condition that follows an autosomal dominant. So autosomal dominant, it is carrying the dominant caused by a dominant allele, which is found on the autosomes. So if we can look at it, that can actually define us that Generation X individual is homozygous. Generation X is homozygous. It can be homozygous, whereas we have got one other one who is unaffected. So this one defines that it is not homozygous. And then Generation Y individual are heterozygous. They are heterozygous Generation Y, which can also, we are saying the answer must be Generation Y individual 3 is heterozygous. Uh, Generation Y, Generation Y individual 3 is what is heterozygous. Generation, uh, we are saying Generation Y individual 3 is heterozygous. I'm sure I remember to say, we say that the homozygous means that they are both the same, which can be either of this. That means to say, or it can be like that. This is homozygous. And then when it is like this, it means it is heterozygous. So what happens here is that number C, Generation Y individual is heterozygous. Generation Y individual is heterozygous. And of course, that can be proven after you can do the calculation here. You can work out this like the way we worked question number 1. And then we can find out that we can have 3 children, one who is unaffected and the other one who is a carrier. And again, therefore, it has to be a heterozygous, which is a carrier of the condition, which must be a heterozygous expression. So we have got it. If we work out individual X, we must Generation 1, which we are saying it can give us two girls, one normal, and then one carrier. So the carrier one is therefore the one who is what? Who is heterozygous. The carrier individual is the one who is heterozygous, as you can see. The heterozygous, this must be the heterozygous expression. So that is what is supposed to be done in terms of this. So it means C is finally correct. We give the biological terms in the following descriptions. Genetic disorders that are controlled by recessive alleles that are found on the chromosomes. We say these are called sex-linked. They are called sex-linked genetic disorders that are caused by recessive alleles. A testable statement that can be supported by scientific evidence, we say that is called a theory. This defines a theory. It is a theory that can be testable and supported by scientific evidence. It is proven genetic growth involving only one characteristic. We say that is monohybrid crossing. Monohybrid crossing. When there are two, we say dihybrid cross. The opposite of this one is dihybrid. Number four, the phase in the cell cycle during which DNA replication occurs. DNA occurs during the interphase. It occurs during interphasing. And the dominance, where the heterozygous phenotype is an intermediate between the two dominant homozygous phenotypes, we call that incomplete. Is an intermediate between, it's called incomplete dominance. It's called incomplete. The phase during meiosis nuclear, when membranes will reappear in two daughter cells. The two daughter cells are within the membranes during telophase. This is it's called the telophase. Telophase 1, and then a change in the organism's nucleotide sequence of the DNA molecule, we say that is called gene mutation. It is called gene mutation. The genetic condition is characterized by absence of blood clotting factor in humans. I am sure you remember the condition of the bleeding woman, which we say is called hemophilia. Hemophilia condition, excessive bleeding because of absence of blood clotting factor. The study of the geographical distribution of species providing evidence for evolution. Evidence of evolution, it's called biogeography. We have got many other sources, DNA. We have got cultural evidence. We have got a video that is actually presented on Metric Hub explaining evidence of theory of evolution. The theory of evolution proposes that offspring will inherit the alleles of favorable characteristics. That is called the Darwinism theory. I'm sure, why do they inherit the favorable traits? Because the unfavorable one have extincted. Because unfavorable traits have died out. So we only talk about the favorable traits being inherited because the unfavorable traits, they have all extincted. The organisms have died, all of them. I will indicate whether it applies to A, B, or none. So cultural evidence to support human evolution, these two, it means it's both A and B. And then stem cells, we are saying undifferentiated somatic cells. Stem cells are actually called undifferentiated somatic cells, which is only multiple alleles. Multiple alleles, we found them in blood groups. it is controlled by three alleles. There are three alleles that controls blood groups, which we are looking at blood ABO grouping. So it is A only. That's what you need to understand here. And we are given representation of protein synthesis. This is one question that I've been answering ever since the channel started, and I want to believe that you guys, you have learned and you can be able to check there and understand. If you go to the channel, you can also be able to testify this to say protein synthesis is one chapter, orogenetics is one topic which has been extensively answered on Metric Hub. Identify structure K, identify structure K, which is actually representing the chromatin network, which is inside, which is covering it. So K is representing the nuclear membrane, nuclear membrane, that is K, and also molecule Q. Molecule Q is the chain of amino acids that are joined together that indicates a peptide, a polypeptide bond. It's called a polypeptide, or this also refers to protein structure. It also identifies the protein structure, that is Q, and then give the letter of the organelle where translation took place. Translation, it takes place by the, give the letter, it takes place by the ribosome. The letter of where translation took place. I want us to understand that translation occurs by the ribosome, which we can see that it is the ribosome, which we say here it is what is O, and tangled network of DNA. Tangled network of DNA, we can see that this is also, if we look here, it is found on tangled, it is tangled network of DNA on the chromatin network, which is L. And then hairpin-shaped nucleic acid, hairpin shaped nucleic acid, which actually we can also see that it is transfer RNA, which is tRNA. It's a hairpin shaped, which is M. Determine the complementary DNA-based triplet for codon P. The complementary base triplet for codon P, if we look at P is this one, which we can give that we have got AAU, so it should also, the complementary, it says the complementary DNA-based triplet. So the bases that, the DNA bases that can pair with AAU, we are saying is, in this case, we are given AAU this, so therefore the complementary DNA basis. So we want the DNA, which we say that this can be, we can have T, we have T, and then obviously A pairs with what? I mean, U pairs with what? With A. So this actually is given as A is the basic genetic, I mean, the basic base pairing that you must take note of. Name the amino acids from left to right as they will bind on molecule N. The amino acids from left to right as they say the amino acids from left to right. So we are saying from left going to right as they bind on the molecule N. So we can have we have got these amino acids. We have got leucine. We have got leucine here. We also have the glutamine, which also is there. So leucine, glutamine is the order that is also, which we can say one and then two. I'm sure this can sometimes comes in a form of a table, which is actually, we should know that we have got leucine, and then we have got glutamine in their correct sequence, which is according to when they bind to molecule N. This is affected. These are affected in the order. These are affected by the order of the what? Of the bases that are on a molecule N. We are saying we are following the order of bases to say 1, 2. So which one comes? A, we say AAU. So we have got also the first one, it must be UUA comes in here. We are saying this side we have got UA, which is the leucine, and then GUC, it would also have, we also gonna have A, G. I'm sure that you should understand where this thing is coming from. CAG. So we are looking on the basis of this. We are looking at the order of this one. These are the ones which we are using to define which one comes first. So we see CCA is the first one, followed by CAG is the second one. So UUA is the leucine, and also UGC, what is it? Sorry, UCAG is what? Is the glutamine, which is that is how we are calculating the order of these. That is where the order is coming from, to say we are looking on the transfer RNA or the anticodons, which comes with their amino acids that are coming according to the order of these ones, 1 and 2. We are looking at N, which has got AAU, and GUC, which is affected in this order. So you see, this order is this order. This order goes up to that, it comes to CAG, it comes to CAG. That is where this whole thing is coming from. That is where this whole concept is coming from. That is where the order of the nitrogen or the amino acids ordering on N is calculated. That is on question number 1.4. Complete the table representing famous scientists, uh, the hominin fossils they discovered at the fossil sites where these discoveries were made. So we need to complete this table there. So we are saying the first one, it says 1.1.5 scientists responsible for discovery misplaced. Misplaced was discovered by Robert Broom. Robert Broom discovered misplaced, and then they named the species. The species name, it was Australopithecus africanus. Australopithecus africanus. Australopithecus africanus was discovered as misplaced and by Broom at Steekfontein Caves. And then we have got Raymond Dirty, named the fossil that he discovered. He discovered Taung Child. Taung Child was discovered uh, by Raymond Dirty, Australopithecus Australopithecus africanus Taung, and then it was in Northwest, and then Karabo was discovered. Karabo was discovered by Berger. They say Lee Berger. Lee Berger discovered this. Lee Berger discovered Karabo, and then obviously the name of the species, it was also Australopithecus sediba. Australopithecus sediba. That is in terms of this evolution that happened, and the table is complete. This, take note of it because it's quite common that it's always appearing in our papers. We are given on 26 December 2004, powerful undersea earthquake triggered a devastating tsunami, claiming the lives of at least 225,000 people across multiple countries. Due to the overwhelming number of casualties and severe damage to infrastructure, mass graves became necessary to bury bodies. People were to be buried, many of them in single grave. DNA profiling was used to identify some of the victims months after these bodies were buried. Some DNA samples were taken from the bodies before burial to compile DNA profiles. DNA profiles were then also compiled from samples provided by family members. These samples were collected from toothbrush, hairbrushes, clothing, and of deceased. The process helped to identify some of the victims. So you can see the situation, and the question is saying, name what tissue can be collected from a toothbrush to use as a source of DNA from the toothbrush inside the mouth. Obviously, we'll get the cheek tissue. We can get the cheeky tissue inside the mouth. The cheek tissue can be found inside the mouth there. Explain how DNA profiling was used to identify some of the tsunami victims. This question is just quite a bit lengthy. I want you to understand that in this case, let me, of course, just remove this one and put it there. We are saying is the cheek tissue that can be found on the toothbrush. So I'm gonna use this space to fill in the answers for next question. So what happens one in terms of question 2.1, 2.1.2, DNA profile bars of victims are compared. DNA profile bars of victims are compared. They are compared to the DNA profiles to DNA profiles. The DNA profile bars from toothbrush or that are also from collected from clothes or every other sources. They are compared with the ones from the toothbrush or clothes, uh, and also samples that were provided by family members. Samples provided provided by family samples provided by family members. And then I want us to understand that if all the bars lines were the same, the person would be or was identified. If all the bars, you can say the stripes were the same, if they were the same, therefore the person was identified. This is a matching process, which we do always on other questions when we come to them to say, explain the possible child or the possible culprit in a given scenario of paragraph. So here, the same thing that you did or that you do when you are given some bars to say these are the bars of the people who are suspected, and then you are supposed to identify, find which one is the perfect match. We compare these. We are comparing the DNA samples with the ones that we have, which are suspects, and then we compare them to see the likeliness of matching. The closer the matching, the higher the chances of person to be related to that sample of the person being compared with. So that is the process which happens with DNA profiling. You are only asked to explain DNA profiling in this case. So in some cases, no personal effects of the victims could be found. No personal effects of the victims could be found, which family members, the victim's spouse or the victim's parents could be used to provide a DNA sample for comparison. We use the parents' victims, not spouse. Spouse, the DNA is not matching, so it is victim's parents. Is the victim's parents which is used. The victim's parents is the one that is used, and it is the victim. We are saying what happens is that the victim will share no DNA bars with their spouses. That is why we say that in this case, explain your answer we are saying victim will not share their DNA with their spouses. Uh, DNA profiles will always, all the bars will match to either the parents of the mother, but they will match to the parents. I think that because the relationship here is actually coming from the what? From the parents. People who are married together, they have no relationship in their DNA because they are unrelated, unlike to the parents who is the father or the mother. That is where the bloodline is coming from. Hence, there can be a relationship. And then describe the structure of a DNA molecule. This is quite a simple question that we always have to understand that we get all the marks. DNA, uh, uh, it is double stranded um, hylax. It is double stranded hylax shaped, which is made up of nucleotides as the building blocks. So I want you to understand that this is where deoxyribose is the sugar. Uh, we have the deoxyribose sugar. We have the phosphate group and four nitrogen bases. There are four nitrogen bases that complementarily pair. They complementarily pair. A pairs with T and G pairs with C. That is what we need to go to understand, and each complementary pair of nitrogen bases is it is joined together by weak hydrogen bonds. Which one, as you write this, the 6 marks are enough and you can get them. The question is, you need to know that DNA is a double helix. It is actually made up of nucleotides, which has deoxyribose sugar, phosphate group, and four nitrogen bases. And four nitrogen bases that are that complementarily pair, which we say that A pairs with T and G pairs with C. Each complementary pair of nitrogen bases is joined together by weak hydrogen bonds. That is enough for the 6 marks that is to be required in this case. Is the phase of meiosis in testicles that is given here, a phase of meiosis in testicles that is given here. a phase of meiosis in testes that is given the questions that are here, identify the phase represented in the diagram, where we have got, we are seeing the phase that is indicated in the diagram. We have got homologous chromosomes, which are aligning at the equator, which we can say this is the equator. And then we have got the, we have the, what to call, the chromosomes that are lining at the equator. So it is actually metaphase. This is metaphase 2. I think in terms of this one, we cannot want to take more time. Meiosis, don't go to exam without working and calculating, understanding all these. A is the spindle fibers. So the spindle fibers give the function of the spindle fibers. They actually control the positioning of chromosomes towards the poles and stuff. Spindle fibers controls the position, of uh, chromosomes or daughter pole, or we are saying position of chromosomes. It affects the position of chromosomes. We can say also chromatids during the process of cell division. So actually they influence, they control, they regulate the positioning by pulling the chromatids. They do pull the chromatids, they pull the chromosome towards the poles or opposite uh, poles when meiosis is occurring. And then in the phase give the number of centrioles. The centrioles, we are saying we have got one, we have also two, which we are saying there are two centrioles. And then chromosome present at the beginning of meiosis in this cell. We have got two of them. So at the beginning, as you can see, we have two there. At the beginning, it means there were four. It's half year. It means this is after meiosis. So at the beginning, the original number was what? Was four. Gametes that will form at the end of meiosis. The gametes that will form also, we are saying it will lead to four gametes forming. Describe the behavior of chromosomes during phase in the diagram above. We say that individual chromosomes from a single row on the equator, we are saying individual chromosome in terms of this. Let me just leave space for next question here. We are saying in this case, individual chromosome, the individual chromosome from a single row they form a single row at the equator. They form a single row at the equator. They form a single row at the equator. Spindle fibers attached to the centromeres. individual chromosomes form a uh, row at the equator. They form a single row at the equator, and spindle fibers attached to the central mass, which is actually metaphase 2, and then explain Mendel's segregation during meiosis. Uh, in this case, Mendel's explain uh, explain Mendel's law of segregation during meiosis. So what happens is that an organism possesses two factors. An organism possesses two factors. It possesses two factors which separate during meiosis. Which separate during They separate during meiosis. And I want us to understand that so that each gamete contains only one of them. So that each gamete contains only one of them. That is the law of segregation of Mendel. Organisms that do have two factors which separate during meiosis so that each gamete contains only one of them. That's what happens during segregation according to Mendel. Uh, we are given that Henry VIII was King of England from 1509 to 1549. During his reign, he had 6 wives. Infamously, he sent two of his wives. It is Anne and Catherine Howard to their deaths on the executioner's block at a Tower of London. One of the reasons he had these wives executed was the fact that they had not given birth to a male, they did not give birth to a male child. So, yeah, that is it. And then, which parent determines the sex of a child? Which parent determines the sex of a child? I'm sure, remember to say, in terms of sex determination, is the father. The father, he carries the distinctive chromosome, which is what? Chromosome Y. Draw a labeled diagram to illustrate the structure of the gonosomes in a male somatic cell. The gonosomes in a male somatic cell, you need to know that we are just looking at a structure of cell, which we have got a long X and also a short Y, which are in the cell. What is important here is the male gonosome here. I can say in this case, this represents an X chromosome. And this represents a Y chromosome also. And then you need to know that this diagram is representing a male gonos. We are looking at a male gonosomes. Male gonosomes. What happens is that you should always know that an X is longer than Y. So the longer one is the X and the shorter one is Y. That is one thing that you must take note of when it comes to sex chromosomes. And then the next question is explain why Henry and his wife was responsible for the sex of his children. Uh, we are saying why Henry and his wife were responsible for the sex of their children. So in this case, what needs to be known is that Henry's gametes have one X chromosome. Henry's gametes have one X and Y chromosomes. I think that what you need to know. Male have got one X and Y chromosome, and and uh, the wives, they have two X. The wives have got two X chromosomes. I think that we know. Uh, so in this case, what you need to know is that if an egg is fertilized by an X-bearing sperm, if a baby girl is formed. If an ovum is fertilized by an X, if it is fertilized by an X-bearing sperm, by an X-bearing sperm, a baby girl is formed. A baby girl is formed if the egg is fertilized by an X-bearing sperm. And again, and when fertilized by Y bearing sperm, a baby boy is formed. That question, what it just wants you to understand is you need just to understand the concept of sex determination to say XX combination. If we have got an X, uh, if we have got an X and an X combined together, they give us XX, which is a female. And if we have got an X times an Y, it gives us an XY, which is also a boy. That's all this question is asking. That's what the question is actually asking. That's all the question is looking for. To say a boy is formed when an X and a Y fuse together. And if it is XX, it means the father has given X, the mother has given X. So for that reason, we're gonna have a female also formed. That is what this question is literally looking for in order for you to express the understanding of sex determination. In all birds, the sex chromosomes of males are of equal length. In all birds, sex chromosomes of males are of equal length and are referred to as ZZ chromosomes. Female birds have sex chromosomes of unequal length that are referred as ZW chromosomes. So we are saying what is understood here is Z. Z is male. And of course, ZW is what is female. That is what is given here. Inheritance of head feathers in sex is sex-linked. Head feathers is sex-linked, and the gene for head feather color is in a bird is only found on the Z chromosome. The gene for head feather color is only found on the Z chromosome. The chromosome does not contain a copy of this gene. Red head feather are dominant over black head feather. So interesting question. So we are given head feather there. So using the letters R and R to do a genetic cross between heterozygous male uh, with red head feathers and a female with black head feathers to show how the phenotype ratio of the offspring will look like. This is quite an interesting one, which we can say that we can say P1, parental 1 phenotype, which is equals to, we are saying the male is red. We are saying red feather and female black head feather. So we are saying red feathered male, red feathered male is crossed with black feathered female. Is crossed with black feathered female. That is the cross that we have. And then we say the genotype for this, we should understand that we are given that using genetic crossing between heterozygous male with red head feathers. So that is to say we said is carried by Z, which is red feathered heterozygous, it is RZ, and then is crossed with a female, which is of course black. We said black is recessive. Black is recessive, which is equals to Z. It equals ZR W. Remember that the gene is carried by Z only. The gene is carried by Z only. So that is it. That is what is happening here. And then we say meiosis. And then we have the gametes that are supposed to come here. So in this case, this one, it can come and give us RZ. And this one can come and give us RZ. And then is crossed with this one. It come down and give us ZR. And this one can come down and give us W, something like that. And then we have fertilization. Fertilization would always occur in terms of the fertilize or the fusion of these. So what would happen is we have got this one. It can fuse with that one. We are going to have we have got Z R Z Z R is this Z and that one. It gives us that one. We have got this Z. It goes with this Z. We have got Z R, this one. And then we have W. We have got this Z. It goes with this one. It gives us ZR. ZR. We have got this Z. It goes with that Z. we have got Z R W as to say these are the possible outcomes that we have, which we can say is the genotype. The genotype that we have got all these four possible outcomes that are produced. So having said this, the phenotypic, so we need to say the phenotype. The phenotype, what is the phenotype? We are saying, if we look at this, when we look at this, we can see that this is red feather. This is red feather. This is, if it is ZZ, it's what it is. This is a male. This is a female. This is a male. This is a female. That is one thing that we can see there. So male with red feather, say male with red feathers, or male with black feathers, male with red feathers. And also what we need to understand is that this ratio, it gives us one male, one male, one red feathered male. Let me say one red feathered, one red feathered male as to this one red feathered female as to one black feathered male as to one black feathered female. That is what is happening. These are one. This is one class. One. This is one. As you can see from the outcomes that we are seeing, that we are having there, one red feathered male is ZZ. It said, we said in this case, we can take ZR, R like this. This is equals to red. That is what we have. RR determines, and we said ZZ. As we have seen here, we have said also that ZZ is female, is male, and ZW is female. That is where the contrast is just coming. So if we have got ZW, originally ZW is female, ZZ is male. And of course, if we are having RR is equals to the same as RR, which is equals to red. And then obviously, if we have got RR is equals to black, that is the only logic that is being asked in this case, because we said this is dominant. It is given that this is dominant. R is dominant over this one. That is what we need to understand. And then if you do this, I'm telling you, your story is just done, and it is well done. That is where the question is coming from. And I hope that you get this right in your exams as we are going next end of the year. Then we are given in a certain type of plant. In a certain type of plant, the genes that determine the leaf shape and the color of the flowers are found on the same chromosome. The leaf color, the leaf could be either ringwood or smooth, and the flower and the flower can be either purple, it could be either purple or red. That is what we are given here. That is what we are given. And then the diagram below represents the arrangement of alleles on the chromosomes in plant A and B. That is what is given there. We are given this chromosome of plant A and chromosome plant B. That is what we are given there. Let me just minimize in order for us to otherwise see it better, so we can apply the questions nicely. So the question next is saying, identify the phenotype of plant A. The phenotype of plant A, what happens is that we take the letters each by each. We have got the phenotype of plant A, we have got T, D, and E, E. So we need to understand what is the meaning of this. We give the interpretation of this. This is genotype. So genotype defines the phenotype. So the phenotype is the meaning of this, which we can see that we are told that dominant D is wrinkled. This dominant D, it means it is given it means wrinkled. It means wrinkled. This is given in context there. It's given that D is wrinkled. We compare this to alone. It tells us that this is wrinkled. And then E, dominant E, it means purple. And then dominant E, this one, it means purple. So the phenotype, therefore, it means wrinkled leaves and purple flowers. Wrinkled leaves and purple flowers. That is what is happening. We interpret them separately. They are together, but in order for you to understand it better, take this one on their own, and then you take these two also on their own comparatively. So the phenotype is going to be wrinkled leaves and purple flowers. Wrinkled leaves and purple flowers. Wrinkled leaves and purple flowers. That is the phenotype of A. That is what we are given there. The genotype of plant B. The genotype of plant B, we are just taking these letters as they are. We are just taking plant B is we are saying this plant B, what is given here is we have got these two on their own and also these two on their own together. We add them together like that. These two, we say this will give us the genotype. That will give us the genotype, which we can see that that can be given as to say, we said GGEE, because genotype is given by combination of letters. I said we take that and that, which is GG, this and that is EE. Together we add them. Together they give us, which is equals to GGEE. And therefore, that represents the genotype that we are talking about. That represents the genotype that we are referring to. And the plant has a chromosome. Identify the plant that has chromosome with the homozygous alleles. A plant that has got homozygous. As you can see, all these are homozygous. So we're saying the plant that has got homozygous alleles, you can see that that is plant B. I'm saying homozygous, you can see they are the same. This is a homozygous. It's a homozygous alleles that we are seeing here in terms of this plant. So it is plant B. It's plant B. And then 2.25, determine how many possible different genotypes the gametes of plant A can have. How many different possible genotypes the different genotypes the gametes of plant A can have. So we are just saying the possible gametes that can be produced from this one, possible gametes which can be produced from this combination. How many gametes can be produced? We have got heterozygous combination, which we say we have got D, and E, E. I always say that what happens is that this is called the random segregation or separation of alleles that we need to understand. So what happens is that we can never have D's in their own gametes and E's in their own. So what happens is we take this one, it goes with this one, it will give us a D, E. If we take this and that, they give us a DE. We take this one. Let me say we take this one. We take this one again. It goes with that one. It gives us a DE, as you can see. So this is the first coil of gamete. This is the second. I can take this one. Sorry. I'll take the second gamete, which is the second one is this D. It goes with this one. We have got DE. I'm taking this one and this one, they go together. And also understand that we can as well take this one, this small d and the small e. They give us a G and an E. I hope you are understanding this. So the possible gametes which we can have, we have got 1, 2, 3, 4. These are the 4 gametes which can be formed out of this. Each of these is supposed to, each gamete must go along with everything in the next. Each allele must go together with each of these, so we can see. That is why you see this G, and we have got a D here, and it goes with the dominant, this one, and that it goes with this. We say this D, it goes with this E here, and then it gives us the first pair. The dominant D, it goes with the small E here, it gives us this one, and then recessive D, the recessive D there, which you see the recessive D, it goes with the dominant E. We have got this one. The recessive D, it goes with the recessive E. It gives us this other one. So we have got 4 possible genotypes. Determine how many possible different genotypes the gametes can have. It can have 4. That I guess you can see the way I have worked it for you there. That is how it works. Just follow the color of the pen that I'm using for you to see how this separation is taking place. And then plant A and B are crossed with each other. What percentage of the offsprings will be ringwood leaf and purple flowers? Plant A and B are crossed This one, I guess you remember to say this combination, we have once worked it before. We worked it before. We got a ratio of 9 s to 3, s to 3, s to 1. If you still remember this ratio of a dihybrid crossing. So we are saying if plant A and B are are crossed with each other, what percentage of the offsprings would ring with leaves and purple flowers, which is this is the one with the recessive alleles. The recessive alleles, the recessive alleles are always giving us a 25%, which is represented by 1. I'm sure you are getting this. If for the interest of time, I'm just not working it, but what you need to understand is you are just going to do a punity square of this cross, which you shall find out that you shall get a ratio which is going to say this one is the dominant alleles, which is going to be it is going to be it's going to be the ratio of this one is going to be giving you the ratio of dominant alleles. The ratio of dominant alleles 9, and then you shall get that the other was 3, 3, and then the recessive one is 1, which gives you 1 over the func multiplied by 100. It gives you 25%. From if anybody still needs more, please just follow and question about it. We can work it together, but I'm expecting that I'm doing term 3 and I'm working with people who have had a good lesson on how do we calculate. And please note that we use a punity square in terms of calculating this. You need to know that you use a punity square because we have got a multiple of outcomes that are there. In order for you not to be confused, use a punity square for you to calculate that this outcome that we are talking about. So I'm sure you count the ones which have got ringo leaves and purple flowers. You shall see that we have 1, and then that will give you a 25%. Thank you so much. I think that has been understood. We have got question 3. A bucado was a large wild goat with long curved horns. For centuries it lived high in mountain range that divide France and Spain. They were once abundant, but extensive hunting reduced their numbers to only 100 individuals in 1899. The last bucado, known as Celia, was found dead in January 2000 by park rangers. Dr. José Foch from Spain had, however, captured Celia the previous year and had taken a tissue sample from her ear. Using the same technique, same techniques used to clone Dolly the sheep, researchers found transplant DNA from the ear tissue into an ova from domestic goats to create 439 embryos, of which 59 were implanted into surrogates' airways. Only 7 of the embryos resulted in pregnancies. This is nice and interesting. So the question which is being asked is, name the biotechnology process used to make identical copy of the bucado. I'm sure you still remember this whole thing of using we use the tissues in terms we say reproductive cloning, which is called cloning. And then why was the tissue sample from cilia Why was a tissue sample from celiac ear used? This is actually because it has got diploid count number of chromosomes contained. It contained the full set of chromosomes. Yes, and it was diploid, which we can say 2n. It actually has the exact required number of chromosomes that we needed. Explain the procedure used to create the exact copy of the buccal using tissue obtained from the ear. This question, class, you just need to explain the process. Explain the process of cloning. Of reproductive. You are only supposed to explain the process of reproductive cloning, which I want you to know that we have got diploid nuclei were removed from the ear as it is given there, and then the receiver cell were egg cells from the domestic goats. The domestic goats are given in the passage that the surrogate was implanted into Airways domestic goats which were there. So what happened is that the nuclei, the receiver cells were egg cells from the domestic goats, and then the nuclei of the ova was removed, and the diploid nuclei of the buccal was transferred into the ova of the domestic goat. Then the ova was stimulated to divide through mitosis, was stimulated to divide first by electrocuting and divided through mitosis. The zygotes developed into embryos, and embryos were implanted into the surrogate goats, which of course the surrogate goats gave birth to the buccal, which was being cloned. That's the process. We are just explaining the process of cloning, which of course you also go to our channel, refer to the channel again, refer to channel. We have got a full lecture which explains this process of cloning. Just look on that process which we talk about, the surrogate mother and also the donor parent, how that process works, saying that the surrogate mother's nucleus is removed and then it is replaced with the diploid cell of the ear in order for it to have the required chromosomes. Then it was electrocuted to stimulate division. After that, it developed into embryo and implanted into the surrogate goats, which gave birth to the buccal. That's what you need to go through the describing how the researchers could have calculated the percentage success rate of implanted embryos that resulted in pregnancy. In this case, what we need to see is that these researchers, we are told that we are given that using the same technique of the dolly sheep, researchers could transplant DNA from the ear tissue into ova taken from the domestic goats, create 439 embryos, of which 57 were implanted into the surrogate airways. Only 7 of the embryos resulted in pregnancy. Only 7. So now what is happening is that they took the 7 embryos that resulted in pregnancy, took the 7, they took the 7 embryos that resulted they say seven embryos resulted in pregnancy. And then divide the number of implanted embryos. Number of implanted embryos. We are given that the implanted, and then we are given that the implanted embryos, which 57 were implanted. We are given that implanted embryos, which were implanted embryos, were 57, 57, and then multiply by 100. Multiply by 100, which is a percentage. So the question just is saying, uh, you are supposed to give the formula. You are not allowed to give a mathematical formula. Don't use a mathematical formula. You are supposed to explain, you are supposed to describe. You describe how this process occurred. 7 embryos that resulted in pregnancy. Divide the number of implanted embryos and multiply by 100. So that is the calculated percentage success rate. Percentage success rate, what came out of what was there, multiplied by 100. That's what you are supposed to do. The diagram shows possible relationships between different hominids. These are possible relationships which are between different hominids, and the question is saying identify the type of diagram shown above. This diagram is called a cladogram. This is called a cladogram. We say it is called a phylogenetic diagram. It is called a phylogenetic diagram. And then how many genera are represented in the diagram? The genera that are represented, we have got the genera, we have got the this, we can say, let me start from here. We have the Homo 1, the plant, we have got the, and then we have got the Pranto 2, we have got Australopithecus something. These are 3, and then the Ardipithecus 4. I'm saying we are looking at these to say we look at them as to say this from the all that are Austro are one thing. All that are Austro are one thing, which is 3, and then the Prantropus is a is also one of its own, which is also represented there, the Homo 1, and then obviously total is 4 with the Ardipithecus. So we have got how many of them? There are 4 of them. Name the species that is the oldest. The oldest species is the one that comes, we are saying the oldest species is the one which is starting, where it started from is the oldest. We have got the oldest. The oldest is Ardipithecus ramidus. Ardipithecus Ardipithecus ramidus. we are saying is the one where the all the generations come from is the oldest, Ardipithecus ramidus, and then name the species that share most recent common ancestors, that share most recent common ancestors with Australopithecus africanus. Uh, the most recent common ancestor with Australopithecus africanus, Australopithecus africanus. If we can just remove these things so we can see, I'm saying, so I'm saying the share the most recent common ancestor with Australopithecus africanus. Australopithecus africanus, if you can look there, Australopithecus africanus, it is, you can see it is the plant. We say this one and this one, they share together the same ancestor, which is Australopithecus afrensis. They all come from this, but the common they share, these two is one, and two, they share the most recent common ancestor. So it is Paranthropus robustus. Yes, Paranthropus robusta. Is it N here? Paranthropus um, robustus. Paranthropus robustus share the common ancestor with Australopithecus africanus. We are moving together, and I guess that is making sense there. And then name two forms of evidence that could have been used to support the information in the diagram above. We are saying evidence of evolution, evidence for evolution, sources of evidence for evolution. We can give this. I also said that uh, we can talk about cultural evidence, cultural evidence. We can talk about the genetic evidence, genetics. We can talk about fossils. Evidence can also be used to explain this, and then give the name of the Homo species that used weapons and stone tools in the first time. I think you remember that this was done according to evolutionary history, is the Homo habilis. They are known of using the stone tools, and describe how the discovery of Homo fossils supports the out of Africa hypothesis, out of Africa theory. This, I think you know that Homo habilis were found only in Africa. Homo habilis were found only in Africa. Homo habilis were found only in Africa. Oldest fossils of Homo erectus and Homo sapiens were found in Africa. Oldest fossils of Homo sapiens and the oldest fossils of Homo sapiens and Homo erectus Homo erectus, and Homo sapiens were found in Africa. And also, younger fossils were found in other parts of the world. Younger fossils were found in other parts of the world. The fossils were found in other parts of the world, and then explain the significance of three changes in human skull to show progression in evolution. The change in human skull, actually, the change to show progression in evolution. One, larger cranium, which is also rounded, increased in size to show an increase in intelligence. Larger cranium, larger cranium actually showed an increase in brain size and intelligence. Showed an increase in brain size and intelligence. So we also have to see that structural score changes. We also have the more forward position of the foramen magnum develop the ability to walk on two more forward position of the foramen magnum actually it indicated a development in bipedalism. Development in bipedalism was shown because of the more forward position of the foramen magnum, which is also a significant increase with reference to this skull. We also need to look at, we also have an S-shaped spine absorbs more. It actually absorbs shock when walking on two legs. S-shaped spine absorb shock when walking on two legs. I can also mention the issue to do with the flatter face. The flatter face, which we say non-pugnacious. The flatter face, non-pugnacious. Also, the development of the chin. development of the gene. We also look at smaller canines, also. We also have the prominent brow ridge, due to softer food and smaller muscles in chewing. Due to smaller, due to softer food, and also uh, smaller muscles for chewing. These are also developmental changes that came with the changes in the skull. The skull changed and then resulted into this, marking a good development in terms of uh, evolution, and also even the development of the gene resulted in development of language. Development of the gene also development of speech. Resulted in development of speech. The gene being well developed, it also, which is a progression in evolution. Right now we talk about languages that are there, considering that many kinds developed from primates, which were not able to talk. Talking came as a result of the development of the cheek. And then we are given determining which type of inheritance accounted for genetic disorders in Dobermans. All dogs have chromosomes count 78. The procedure used was as follows: genome 1,200 dogs, and all the dogs suffered from various genetic disorders. To ensure all that desired characteristics are present, breeders used the process of artificial selection. They drew sample of 5 liters of blood from each dog to collect the necessary genetic information. That is the result of the investigation is presented in the table that is given here. So, identify the independent variable. The independent variable the scientists investigated determining which type of inheritance accounted for genetic disorders. So what we are saying is type of inheritance. Type of inheritance is actually an independent variable. Type of inheritance is actually independent variable. Type of inheritance is actually the independent. Two ways in which the scientists ensure the validity of the investigation. We can see that they used the same type of dogs, used the following. They used the same type, they used 1,000 dogs of the same breed were used, and also they also drew a sample of 5 milliliters, from each dog for genetic information. So we have got to understand that they use the same breed of dogs, use the same breed of dogs, and also a big sample was used. A big sample of 5 milliliters. The sample for the genetic information was used 5 milliliters, and then explain why there is no need to keep the Doppler's dog age constant in this investigation. Uh, the reason is that we are to work on genetic effects, so genetic conditions are not affected by age. Genetic conditions are not affected by age. I need you to understand that they are not affected by age, but they are inherited, uh, and then they are born with the condition, but they are inherited. Dogs are born with the condition. They are they inherited the condition. They inherited the condition. They inherited the dogs in, but they are inherited. Dogs are born with the condition and are affected for their entire lifetime. They are affected for the entire life. So genetic conditions are not age dependent. These are inheritable conditions which are affecting organisms, and therefore they can have them for their entire life. And then we have got explain why the inheritance of autosomal dominant disorder is so much higher than the inheritance of sex-linked disorder. Uh, we are saying autosomal dominant. Why is the inheritance of autosomal dominant is higher than inheritance of sex-linked? Uh, what happens here again is that I want you to understand that the allele for genetic disorder uh, will mask the effect of the allele without the disorder. Number 3, 3.34. Let me just put it here. 3.3.4 is just to understand that allele for genetic disorder, allele for genetic disorder, the allele for genetic disorder will mask the effect of the allele without disorder. Will mask the effect of the allele without the disorder. That is the simple terms which I want you to understand that alleles for genetic disorder will mask the effect of the allele without the disorder. So we are supposed to see this would also cause uh, autosomal genetic disorder can occur, and we have got only two chromosomes on the sex-linked genetic disorder that can occur. I'm sure you still remember this to say uh, sex-linked, we only have two alleles, and then autosomal, there are two men. So question number 5, 3.35, 3.35, this one. Uh, it's just a similar question that we calculated in the assignment that was given to say draw the pie chart for this question. In order for you to draw the pie chart, you are just supposed to convert this into a revolution. Draw a pie chart, draw a pie graph to represent the information provided in the table. The information provided in the table, but in this case, we are already given in percentages, so we can still draw the graph without calculating, having this one as the highest pi, and then we have got 711, the second, we have got the third pi, and the smallest, because we are already given this as it is there, so the pie chart can be drawn without calculating the degrees, because we are able to draw this based on what? Based on the percentages that we have. So based on the percentages that we have, we can draw. The biggest one is we have got 1, 2, 3, 4. So we have got 71%. It must be big. 71%. Let me see if I can say 71%. Say 71%. 71%. And then we have got 11%. We have got 11%. And then I can say we have got 8%, and then 10%. Therefore, we can be able to draw this pie chart using percentages, unlike to calculate them again, because we already have our information converted into percentage. It can help us to calculate or to draw this pie chart with easiness. We can draw our pie chart with ease because it is actually possible based on the percentages that we are having. We are able to work it out and get these answers as we can need them. So having this, we can see that this is the pie chart that is needed there. This is the pie chart which we can give there to say autosomal dominant, autosomal dominant. We have got other, and then we have got sex-linked, and then finally we have got the autosomal recessive. We have the autosomal recessive, which is actually drawn in that case. Our graph is actually done. Our graph is done based on percentages because the percentages are there. We can see the variation in the representation of this. So yeah, class, that is it. That is the question. And then maybe the last question. The last question which is given is actually given here. Use an example in the passage and explain. Use an example in the passage and how dog explain how dogs initially may have evolved through natural selection from an extinct group of wolves. So you are only explaining natural selection. Explain natural selection, survival of the fittest. That is where the question is coming from. Explain why all breeds of domestic dogs belong to the same species. This domestic dog compared to the wolf produce much more easily, allowing dogs to benefit from high-starch diet of humans in a way that the wolf would never can. So what is happening is that I'm saying it's a question which is actually given for us to, like the way we have seen, we've answered all these questions, and you can see that they are actually the same. Why all breeds of dogs belong to the same species? They evolved from the same ancestor. They evolved from the same wolf ancestor. That is why number last, they evolved from the same ancestor. They share a common ancestor. They evolved from a common ancestor. They evolved from a common ancestor. Therefore, they have the same species. Class, thank you very much for your time, and all the best in this upcoming term and also exams that are to come. Keep following, and let's keep doing it. Here is the time and it is now. Thank you very much.